Neurosurgery and the director of her laboratory. She completed her initial training in Armenia and then in France, and then came to the United States for a research fellowship at Memorial Sloan Kettering before completing an MBA at Emory and beginning her research career there. She is a widely regarded researcher in the field of neuro-oncology, studying the tumor microenvironment, particularly in glioblastoma and other high-grade gliomas like DIPG. And I'm pleased to welcome her to present some of her research today.
Thank you very much. And I'm sorry, guys, I'm going to have to be Frank's beautiful talk. Now I'm in thought process myself, so I have to bring back you into research. We are not surgeons, but we are mouse surgeons doing a research on brain tumors in mice. So, and let me see if my sharing is going to work. Yeah. Now. Okay, I think this is going to be okay now. Give me 1 second. Perfect. And so today I'm going to talk about interleukin signaling pathway, but specifically because my lab studies tumor microenvironment, and IL-1 is one of the potent pro-inflammatory signaling pathways. And although it's so known, it's not very well studied in GBM context. And it's a signaling pathway that connects brain tumor microenvironment and tumor. I think as an example, it's not the only one, but today I'll focus on interleukin signaling pathway. So I have nothing to disclose. And before I start my talk, I want to thank very much my laboratory members that did the work that I'm going to present today, especially Zi Chang and my former graduate student, Cameron and James, that did most of the work, and my current lab members that are keeping the project going. I want to thank to my collaborators, especially Alex Tsankov and his postdoctoral fellow here at Genomics, Bruno, that introduced us to single-cell sequencing and the power, how we can dissect cell type specific effects. And Nadia, who is a neuropathologist, been very instrumental providing us patient samples that we can validate our discoveries in mice to human, and my collaborators at Emory, Dima, and David Katman from FASU. So before I go, I just want to introduce you the Cancer Genome Atlas that over like 15 years ago divided glioblastomas into, based on RNA expression profile, into preneural, classical, and mesenchymal subtype. So these subtypes are not mutually exclusive. They actually can coexist within the same tumor, both in a single cell level and also in a regional level. But those subtypes basically represent the dominant transcription pattern at the time of tumor resection. Later in 2016, the WHO divided glioblastomas into clinically meaningful subgroups, not based on expression, but clinically meaningful, divided them into IDH1 mutant tumors that have a better clinical outcome compared to IDH1 wild-type tumors. So it'll be fair to say that IDH1 mutant tumors are completely different entities with better clinical outcome, while IDH1 wild-types can be divided into preneural, classical, and mesenchymal, but the survival is not different between the subtypes. When you add the genetic heat map into this, because modeling for us, the RNA expression profile, when you have 1,000 genes up and down, it's very difficult. But when you use genetic alteration, you at least can model that in mice. And if you add the heat map into RNA expression profile, you can see that mutational burden in GBM is low. This partially can also explain low level of neoantigens and failing immunotherapies in glioblastoma patients. But if you carefully look, although there are tumor suppressors like CDKN2A, P53, P10 that are common tumor suppressors, their losses or mutations are uniformly distributed among preneural, classical, and mesenchymal subtypes. But if you zoom into signaling pathways like PDGF receptor amplification, EGFR, especially V3 mutation, which is the most common, and then you look at NF1 gene deletion, you can see that although not mutually exclusive, they seem to be predominantly associated with a certain subtypes. And that's the information we generally use to model in mice. But then in 2019, this was the TCGA for 15 years doing this. And then 2019, the paper from Harvard group, from Mario Suva's group, came saying, well, you know, we don't have the subtypes, but now because GBM represents, each sample represents mix of cellular states. And these states are like OPC or associated cell of origin, OPC, MPC, AC, or mesenchymal-like cells. So you have a tumor that represents 4 cellular states. Yes, there is some association with cellular states and genetic alteration. Like example, OPC cellular state is associated more with PDGF receptor amplification. NF1 alterations are more mesenchymal cellular state. And from all these subtypes of cellular state, mesenchymal seems to be more driven by tumor microenvironment, tumor interaction. So to model, and I'll get back to both systems, to model in mice the disease in human, which is not very easy task, we use RCAS TVA system, which was developed by Eric Holland, who is a neurosurgeon scientist at Fred Hutch now, and when he was a postdoc in Herald Pharmacy's group. So the system is very simple. Use RCAS virus. It's a chicken virus. We don't have mammalians, don't have receptors, so we are pretty much not infectable by RCAS viruses. So we use it as a delivery system for silencing of tumor suppressors or overexpressing oncogenes. So you put here short hairpin for tumor suppressor or oncogene, and then so normal mice, they are not infectable. But we have genetically engineered mouse models that we specifically express the receptor for this virus in cell type specific promoter. In this particular case, over my talk, I'm going to talk about nesting TVA mice. And in adult mice, nesting labels stem progenitor cells that are also thought to be cell of origin for glioblastoma. So then we use surgeries, not as sophisticated, obviously, in a little mouse brain. And based on the region that we are targeting, we can put our virus-producing cells there, and they will infect nesting positive cells. And what they will do, they will create de novo GBM in mice. And we use this system successfully to generate adult glioblastomas. And there we use adult GBM-specific driver mutations, and we use the same system for a pediatric subset. So we do all histone mutant, DIPGs, or cortical GBM in kids. So the tumors that are generated with this system, they closely resemble, this is MRI images of murine and human GBM. And then when you look at the histological features, they nicely recapitulate cytopalisading necrosis. You can see the structures and microvascular proliferation that we see in human GBM. So imaging and histological properties are similar. Then when we did the expression profile on these tumors by bulk sequencing, then you can see that PDGF-driven tumors clustered together, which is not surprising because of oncogenic driver is the same, NF1s together and EGFR V3s clustered together. Then when we said, well, okay, we use the driver mutations that are also seen to be predominant in corresponding human subtype. If we plot them against human subtype, how well they will cluster with their human counterparts. As you can see, PDGF-driven tumors are clustering with preneural, which is not surprising, EGFRs with classical, and NF1 loss tumors are closely clustering with mesenchymal GBM. But that's on the bulk sequencing, and predominant expression profile is similar. Then we looked what was published in 2019 with the cellular states, and we wanted to see whether these cellular states seen in human, we will be able to recapitulate with our mouse models. And this was done by talented postdoc Bruno, who plot the data against human, as you can see. So PDGFB tumors, which not surprising like human, which is OPC-like state, is associated with a PDGF receptor amplification. Here in PDGF-driven tumors, you have more OPC-like cells, but you do have all the variety of cellular states that you see in human in a mouse model. What happens when you have NF1-driven tumors? There, you have more prominence of mesenchymal-like cells. So if you do a bulk sequencing, it won't be surprising that these tumors in PDGF will cluster with preneural. When you do this, because the predominant cells in this subtype are mesenchymal cells, obviously they will cluster with mesenchymal subtype. So that gave us some confidence that these models will be useful tools to study tumor, cell, and immune microenvironment interaction. And it's essential for this particular case to have this when species are the same. So we don't have human tumor transplanted in immune-compromised mice, because there we are neglecting the entire adaptive immune system. And we also demonstrated that innate immune cell infiltration is also affected in PDX models. So the choice is left for us is to use genetically engineered models to study immune cell interactions. But when you look at the bulk sequencing level, you can see also that the inflammatory pathways here, so PDGF tumors are more cold for inflammatory pathway, but mesenchymals are more hot. And that's exactly what was shown in human disease when they show that mesenchymal subtype is more associated with macrophage gene signatures. So with this in mind, we did a basic experiment, and this is years of experiments to do, which is in some cases difficult to do in human, but now with single cell sequencing, it's possible. When you dissociate the resected tumor, you can see only 65% of cells from the tumor mass are tumor cells. Then the rest 35% of cells constitute tumor microenvironment. So it's a close interaction between tumor cells and tumor microenvironment that leads to GBM progression. And the most abundant cells in tumor microenvironment, and the reason we started targeting these cells, are tumor-associated macrophages. So today, during my talk, I'm going to talk about heterogeneity within the same tumor. So intertumoral heterogeneity is another layer of complexity I'll leave for another talk. I'll focus what happens within the same tumor, what kind of heterogeneity we have within the same tumor. So just before I go and introduce tumor-associated macrophages, it's just a one term, but actually it has mixed population of cells. It has the resident brain innate immune cells called microglial cells, which come from yolk sac progenitors early during development. That's embryonic day 9.5 day, and they stay there. And microglia in a resting state looks beautiful like this. And when they get, if any injury to CNS, to be traumatic brain injury, MS, or other diseases in CNS, microglia gets activated. So those cells are genetically stable, but then when they get activated, they can change their expression profile. And it largely depends from the injury or disease context they are associated with. In another, and that's what we have in CNS, healthy CNS. That's our immune cells in CNS. Then in the periphery, we have hematopoietic stem cells that through progeny give rise to monocytes in bone marrow. And then monocytes, when it's formed in bone marrow, exits bone marrow, get into the circulation. And we have two types of monocytes. We have inflammatory and circulating. And if there is no injury or inflammation in the body, in a healthy mouse, for example, these cells are short-lived, very different from microglia. And then they get constantly replaced by hematopoietic stem cells through all the progenies. But when there is an inflammation, in the case like CNS, when you have a brain-blood barrier is compromised, you have leaky blood vessels in GBM, what happened that inflammatory monocytes exits the circulation, get into the brain, and once it touches the brain, it gets differentiated into tumor-associated macrophages. And at that moment, it becomes indistinguishable from resident microglia. And these two cells together, they compromise up to 30% of tumor mass when you dissociate the tumor into single cell. So my lab's point was to study how, first to distinguish two cell population, to see how much blood circulating cells contribute to tumor progression. But we figured out whether this is the MCPs, monocytes chemoattractant protein family, which is the members that are essential for monocyte exit from bone marrow to circulation. So tumor hijacks that mechanism, increases the expression of this chemokine to attract monocytes from blood now to go into the GBM. And we had to develop tools that we could distinguish these two populations. And I'll tell you why is it important to distinguish, because in the tumors, those are localized in different locations. Microglia is localized mainly in invasive age and promotes tumor cell invasion in a invasive age in the margins. And you have macrophages that are localized within the tumor, and they promote tumor growth. And they produce IL-1, and I'll introduce that later on. So first, we wanted to understand from blood, these cells come from blood, how early during tumor development this comes. So we did initiate tumors, and this is little mouse brains. Here you can't see the tumor because size is smaller, and then you have tumor sizes a different variety of sizes. And that's where the tumor, you can see with W intensity here, that's where the tumor is localized. And these cells are the cells that are red that come from circulation. And green cells in the peritumoral and the brain parenchyma are microglial cells. You can see independent from smallest to the biggest tumor, you have these cells coming. So these cells come very early during tumor development and promote tumor growth. When we analyzed these cells and isolated and normalized against their controls, like macrophages against their progenitors in the circulation, monocytes, and microglia activated in tumor context to microglia naive, you can see, although they share like 530 genes, there are significant differences in their expression profile. And when we, okay, hold on a second, I think. Yes, so we wanted to understand how fast the differentiation process of monocytes goes. And it's very difficult to do in histology only because you have a snapshot of a moment and you don't see how long it takes the cell to differentiate. We use two-photon confocal microscopy, which we basically do cranial windows. When mice emerge, we make the tumors very superficial. And when the tumors emerge, we do surgery, we cut the skull and we glue the window, and then we can put the mouse under the microscope. And this is the dextran red that shows the vasculature. And this arrows here, the cubes here show peritumoral area and inside of the tumor. And we can zoom and we can image. We label the cells, which is easy to do in mice, one color or another color, and we can trace them based on that. If you look, this is just a naive section of bone marrow reconstituted mouse, which microglia is green, uniformly distributed. There are about 13% of cells in CNS. And this is in the tumor context, the microglia look. So the microglial cell seems to be in a peritumoral area activated. And when we image them, they extend and retract their images. And this is imaging of microglia movement for hours in a live mouse. You can see they extend and retract their processes, but their body doesn't move. So this might explain what happens when tumor grows. It pushes microglia in a peritumoral areas, because microglia cannot move. The reason we see bone marrow derived cells within the tumor, because they come from circulation and they occupy the tumor, and this process is ongoing process. And this is a bone marrow reconstituted mouse. So now macroglia you can't see. And any green cells that you're going to see now in the next slide are cells that come from bone marrow. And this is the tumor. And you can see this is where we initiated tumor. And you can see the white matter tracks. They migrate towards opposite hemisphere. That's how infiltrative they are. But anywhere you see even them infiltrating in a small nodules here, you can see them surrounded by bone marrow derived myeloid cells, macrophages. And when we look at their movement, you can see that the DMGFB cells are newly infiltrated cells. They are so dynamic, and it's not surprising because these cells are dynamic cells. But the moment they differentiate into macrophages and they look like microglial cells, they also extend and retract their processes, but they don't move. So this can explain why a majority of these cells are localized in the perivascular areas, because the moment they infiltrate, they quickly differentiate. And macrophage is a stationary cell, it can't move much. They become stationary in the perivascular areas. So we wanted to understand whether this is a compromised brain-blood barrier that contributes to infiltration of the rarest population in the circulation, to GBM, or it's the chemokine gradient that tumors hijack that mechanism to bring them into CNS. So we wanted to address both questions. And to do first, we did with the normalizing vasculature, which we do with bevacizumab analogs from Genentech. It's called B20. So we generate the tumors, and then we give B20, which is bevacizumab, which normalizes the vessels, basically decreases the leakiness of vasculature. As you can see here, the shape is very different. This antibody treatment, you have massive infiltration of these cells, but here you have much less cells and they are more differentiated. So this says that vascular permeability is contributing to monocytes, increased monocyte infiltration. But as you can see, we barely reach significance in decreasing their number, suggesting this is not the only mechanism to bring the monocytes. And it wouldn't be, because it still requires multiple steps for monocytes, even with compromised vasculature, to get into CNS. So then we wanted to see whether it's the chemokine gradient. So for monocytes to exit bone marrow and get into circulation, they need a gradient of monocytes, chemoattractant family proteins that are 4 members, both in human and a mouse. And they are called either MCP 1, 2, 3, 4, and 5, human and mouse are different, or they are called also CCL 2, 7, 8, 12. Interestingly, they are localized right next to each other. Nature is very interesting, and that says that it's going to be a redundancy in function, meaning if you target one, the other will compensate. And similar in different chromosomes, but in a human as well, they are localized right next to each other. So we've then taken an approach that we start targeting one, second one, third one, fourth one, and unsuccessful because we had the compensation with other chemokines kicking in. So the target was to remove entire this segment from chromosome 11. So we did that. But today I'll leave it, and we are finishing the story too. So we cut using CRISPR-Cas9 editing technology. We removed the entire segment from chromosome 11 in a mouse, and all 5 chemokines were removed. So we were able, with this mouse, all I tell that, we were able to trap the monocytes in bone marrow. They couldn't get into circulation, therefore they couldn't migrate to the tumors. But at the same time, when we're doing this strategy, we're looking at something that is upstream of this chemokine and might regulate them. And that's where we stumbled into interleukin-1 beta. It's a very, very strong cytokine, very potent inflammatory molecule that it's in a mouse. It's cleaved into its active, it's produced as a pro-IL-1, and many cells can produce and express RNA for interleukin beta, but only monocytes and myeloid lineage cells and neutrophils can cleave pro-IL-1, forming an inflammasome complex into biologically active IL-1. And that IL-1 can be secreted into environment, and tumor cells actually have the IL-1 receptor, and they can respond to myeloid-derived IL-1 by activating NF-kappa B pathway, which is a major pro-inflammatory signaling pathway. So we went to the IV Brain Atlas, where they have a laser capture microdissection done in human GBM patient samples, and they can basically tell you where the message is coming. And if you look at an IL-1 pathway, you can see IL-1 members are either in the peri-necrotic areas or in the perivascular areas. And when we look at the cancer genome atlas, we used like 373, I think, GBM patient samples, and we stratified them based on RNA expression for IL-1 into IL-1 high, and here you can see high expression, and IL-1 low expression patients, and then plotted that against the survival of the patients. You can see that high expression of IL-1 beta is associated with significantly shorter survival of patients. We didn't see, although there was a trend, actually it's significant for IL-1 alpha. So this is the other ligand for IL-1 receptor. Then we wanted, okay, this is only one single gene. There are other covariances there in a patient sample correlative data that can affect the results. So this is the complex IL-1 pathway on all downstream and upstream targets. And when we plotted major players in the pathway, stratified the patient based high and low, we see that this holds true from the top to the bottom of the pathway, meaning the pathway really matters in human GBM. Then we did patient sample stainings for IL-1 beta, as you can see, and we did the peri-necrotic areas too. You can see that IL-1 is expressed by IBA1 cells, which are the IBA1 is a pan-macrophage marker, and this is in the perivascular areas. But then we also got patient samples from Nadia when we did ELISA for IL-1. You can see IL-1 levels are increased in GBM samples compared to control samples. So we wanted to understand how tumor cells induce IL-1 expression in myeloid cells. We can do bone marrow, primary bone marrow cultures from the bone marrow of BLAC6 mouse, and we drive their differentiation into macrophages, and then we co-cultured them with brain slices. And right away, we noticed that it induces RNA expression, both IL-1 alpha and IL-1 beta. It means tumor cells induce IL-1 expression in myeloid population. And then we did IL-1 beta ELISA in mouse samples, where we have a lot more samples that are genetically defined. You can see that IL-1 is upregulated in PDGF, but it's much higher expressed in mesenchymal GBM, similar to what we see in human as well. Then to determine the biological significance of IL-1 in GBM, what we did, we got an IL-1 beta germline knockout mouse. So mouse doesn't have IL-1 beta. We crossed them into our TVA transgenic mice, and we got the wild type for IL-1 and IL-1 knockout. And we induced de novo GBM formation in mice. You can see similar what we saw in a human survival. Mice that have IL-1 betas live significantly shorter. And this is purely driven by microenvironment, because tumor cells cannot process IL-1. So the source of IL-1 here, it's the microenvironment driven. So this is a significant effect on survival, and it's specifically driven by IL-1, which comes from myeloid cells. Then when we look at tumor tissue for phosphohistone H3 staining, which labels M-phase cells in cell cycle, especially in M-phase, you can see that loss of IL-1 results in decrease of proliferation of tumor cells. We wanted really to confirm that in our tumors, in a wild type, we did the single cell sequencing. This, you can see how large is the myeloid contribution when you do versus the tumor cells, and lymphoid is very low, which is the case in human tumor as well, and then you have the stroma. When you look at RNA expression levels, you can see that IL-1 beta, both, and IL-1 alpha expression is specifically localized to myeloid compartment, and the RFP is our marker that will label our oncogenic driver. This is our tumor cell population. It's true, so signal comes from myeloid cells, and responding cell is the tumor cell. And this is just to illustrate the heterogeneity of myeloid compartment that you can see. You have various myeloid subset and dendritic cells, and this is the heterogeneity within tumor population on single cell level. When we look at the changes in a myeloid compartment, you can see as predicted, we have dose dependent. So we lose one ligand, IL-1 beta, and then we lose second ligand. So we have a dose dependent effect on IL-1 receptor. We see dose dependent decrease in monocytes infiltration. Next, we wanted to validate, which is the case, whether with IL-1 beta loss, we're losing infiltration of inflammatory monocytes. So this is the T-SNP plots of like 25 color FACS analysis that divides the cells based on surface marker expression into their subset. You can see here the CD45 and CD11B population. It's the immune infiltrates, both adaptive and innate immune cells express CD45. And then CD11B distinguishes the T cell and adaptive immune cells from the myeloid population. And you can see there is no changes in microglia, which is not surprising because the monocytes, chemoattractant proteins, and IL-1 are not involved in a microglia migration, but you do have bone marrow derived myeloid infiltration is significantly decreased. And this is mainly due to inflammatory monocyte decreased with no changes in neutrophil levels. Then we wanted to know, okay, IL-1 comes from myeloid cells. We see significant effect of survival in tumor-bearing mice. We see decreased tumor cell proliferation. What happens when we add IL-1 to tumor cells? And it's our primary tumors can be easily dissociated, and we can culture them in a dish, and we can do all kind of biochemical studies on it. As you can see in a control, when we culture without IL-1, the expression of monocytes chemoattractant protein family, which is responsible for monocytes recruitment, is actually low. But once we stimulate with IL-1 beta our cultures, we have massive induction of all chemokines here. And that's true also on a protein level when we do ELISA in the cultures. So one way that IL-1 induces increased infiltration of monocytes is increasing monocytes chemoattractant family protein levels to recruit more monocytes coming from circulation to the tumor. And then how the mechanism, how that happens, we looked at NF-κB pathway activation. As you can see, stimulation with IL-1 leads to NF-κB pathway activation in tumor cells. That in turn, NF-κB induces MCP family chemokine expression by tumor cells. When we look at what happens in tumor cells themselves, so we did a stem cell assay. This is when you see glioma stem cells, which been associated with therapy resistant. This is population of tumor cells. And you can define them in a mouse by functional assay, which is called Hook's dye exclusion assay. You give the dye, it's a stem cell assay adapted from stem cell field. If you give the IL-1 beta, you can see that you increase this population SP that represents stem cell population. When you add IL-1, the stem cell fraction increases. So meaning these cells, you don't get Hook's dye in these cells. That's why they excluded. They have high level of ABCG transporters. So IL-1 stimulation increases stem cell frequency in mouse models. To determine whether this is also happening in human, we use human patient-derived glioma stem cell cultures that have a preneural signature, which corresponds to the mouse PDGFB-driven tumors. And we did limited dilution assay. When you take and do dilution of the cells and look at the colony formation, you can see that when we have a control, when we treat it with IL-1 beta, frequency of stem cells in human cell culture increases also. And if you use IL-1 receptor antagonist, which binds to IL-1 receptor and then doesn't allow IL-1 beta and all IL-1 alpha to bind, you can see that you push the frequency, you have much less stem cells there, both in both lines in human. Then we wanted to see whether this is all in vitro that we showed mechanistically. We wanted to test whether this happens in vivo also, whether it's true that IL-1, if IL-1 directly regulate MCP family chemokine production, then IL-1 beta, alpha and double knockout mice, they should have decreased level of these chemokines. And that's what we did. We did NTVA, IL-1 alpha beta wild type mice and knockout mice. We generated tumors, and when tumors emerged, we did ELISA on samples. And you can see chemokine family members, they all decreased, and we yes VEGF-A because it's not known to be IL-1 target as a control that there are no changes on VEGF levels. But then we wanted to do pharmacological targeting of IL-1. We did proof of principle this, and we did surgeries again in mice. And here we put the cannula, and then it has a little top on it, and you can open every day with transplanted driver. The bar hole of the tumor initiation was, like you can see in the picture. And you can do all every day, you unscrew the top, and you can add the IL-1, either anakinra, which is IL-1 receptor antagonist, or anti-IL-1 antibody every day, directly delivering to CNS, directly to the tumor. As you can see, this is an IBA1 staining, and you can see with IL-1 receptor antagonist or anti-IL-1 beta antibody treatment, you have significant decrease of macrophage infiltration, which is quantified here, and you can see that pushes survival of tumor-bearing mice, showing that pharmacological targeting of IL-1 has a potent anti-tumor effect in tumor-bearing mice. I want to say the effect is not as significant as you've seen with the germline knockout, because this is a local delivery we're doing into the tumor. So the diffusion is like probably you get 50% coverage of tumor, not complete. This is just one. We also showed that IL-1 a couple of years ago also is associated with tumor-associated edema. So you can see that it's basically the water content with the crude assay. When you do water content in tumor, you can see with dexamethasone, which we use in a clinic to decrease the edema. The same effect we see in IL-1 alpha beta double knockout mice, and we demonstrated that IL-1 beta is actually downstream target of dexamethasone. That's how dexamethasone regulate brain-blood barrier and vascular permeability and edema. And you can see in tumors that have less with both dexamethasone in IL-1 alpha beta double knockout mice, when they have less water content, mice can live longer because tumors can grow, because edema, if you don't control, will kill tumor mass, will kill the tumor. So you have reverse correlation here. So I think what happens with IL-1, basically we have bone marrow that produces monocytes, and there is an MCP gradient that allows monocytes based on the gradient to get into the circulation. What happens when you have a tumor in GBM, tumor cells hijack the mechanism of MCP production. They start producing MCP and bringing macrophages in the tumor. And macrophages produce IL-1 beta, which in turn signals back to tumor cells to increase more MCPs and bring more monocytes into circulation. So IL-1 basically, and in addition, IL-1 also, which I didn't talk today much, but IL-1 also signals to endothelial cells and leads to NF-kappa B pathway activation there and increased permeability, which contribute to vasogenic cerebral edema formation in GBM. I think overall, it seems that IL-1 can be good target for GBM. It can have anti-tumor effect, and it will have anti-edema effect in GBM patients, and at least in a mice. So, and I think I finish with this, my talk, and I'm happy to take questions.
Hey, Dolores, that's an awesome talk. My video is not working for some reason, but so everybody, Dolores is really one of the well-known experts in microglia and myeloid cells associated with GBM. So the tumor microenvironment has become kind of a very heavy focus for study of GBM, as you just heard, and what the story that's developed over the past few years under Dolores's leadership is really the microglia are a major component of tumor development, and targeting those cells, which are the most numerous cells, has become a real focus, a real targeting paradigm shift. And I think, so one of the questions I have for you, Dolores, and I know we're trying to focus on the pathways associated with the microglia, but why not use the microglia almost like a Trojan horse, right? So the microglia are so numerous around tumors, why not try to convert them into an anti-tumor type Trojan horse? So, they go there, they're attracted to the tumors, as you've shown, but switch them into an anti-tumor role where they can attack the tumors. I'd love to hear your thoughts on that.
Yeah, thank you, Kostas. It's a great question. So it has been done. People have been trying with that would happen with CSF1 receptor inhibitor. people have been trying to change these cells from pro-tumorigenic to anti-tumorigenic, but these cells, they are genetically stable, right? They don't have mutations, but when they get activated, they just behave. Targeting them it's been failing. So initially, CSF1 receptor inhibitor was successful, but then they developed resistance to it. So you target one pathway, they induce other survival pathway. So it's been challenging. People have been working on it, and it's a challenging task to do because they evolve with tumor progression. Like neutrophils and other myeloid subsets, been shown they come early and they are anti-tumorigenic, but very quickly they convert into pro-tumorigenic. And switching in a progressing tumor that these cells constantly change has been a challenging to do. This is why I think right now we are working on blocking their infiltration. Some reason we took this approach because we are like, when they are there, it's very difficult to manipulate them because they develop very quickly resistant mechanisms. While targeting them, maybe it'll be better because if we block their infiltration, then they won't go there. They won't support the tumor growth. But then you get another compensatory infiltrations coming from neutrophils. So it's a challenging task, but it's work in progress.
Yeah, exciting work.
Yeah.
I also love this talk. What was the time course of watching those green cells migrate? How fast was that happening? The second slide where they were still not differentiated.
So this was a condensed video. So we condensed 6-hour video to the speed you show. It's very fast.
I see.
But I think it's an extremely fast process of transmigration then from circulation into the tumor. It's pretty fast process. But we image them for days. We can image them. They live perfectly fine if you keep them separate with the cranial window. And it's nice because you can also give a therapy like we gave anti-VEGF antibody. We completely changed the vascular permeability and look at their dynamic infiltration changes. We're planning to do that with radiation and temozolomide. As well.
You know, the other question I was going to ask on piggybacking on Kostas, it seems that, glioma is the invisible enemy, and differentiating tumor cells from non-tumor cells is essentially the difficult part of treatment. But you've demonstrated a mechanism by which microglia know where the tumor is.
Yes.
And so even if you can't make those the killer cells, can you use the same sort of signaling to attract some other type of killer? I mean, how is it that those microglia know to go there? It's very hard to differentiate tumor from non-tumor. So they're pretty smart cells, aren't they?
Oh, they are yes, they are very smart. And once they get in a communication with tumor cells, the connection is so strong between, they feed into each other and they support each other. So yeah, it's a good question. It comes down again, targeting them has been very difficult. Yes, targeting. So I'll tell. So we did this mouse that we removed all the chemokines. We trapped them in a bone marrow. Now they can't come. We got compensatory infiltration of neutrophils.
I see.
And it wasn't the Z in my lab who did it. It took 2 years to make that mouse and validate it. And we were expecting we will block major monocytes infiltration to tumor, and we're going to probably extend the survival dramatically. And it was surprisingly because we didn't see difference. When we decrease the dose, we see difference. When we abolish it completely, which is the goal to abolish it completely, we saw compensatory neutrophil infiltration. So we had to go back and now block neutrophil infiltration, which is not an easy task either. But now we're doing experiments where we block both. We'll see which cell will come. Yes, it's a good point. And hope it's T cell, right, that comes. But I can't say now, but that's what we're working on.
You can certainly see, the benefit of having you here, and the fact that you came following Kostas from Emory. It has been a very fruitful period, and really nice to see your lab flourishing like this. I'm glad that you're here. I see there are questions from Trevor and Alex.
Thank you.
Hi, thank you, Dr. Hambardzumyan. That was an awesome talk. I had a question related to the microglial motility. Given that the tumors themselves are increasing IL-1, IL-1 beta, and it's been shown in certainly other pathologies that interleukins increase microglial motility, the very nice video that you showed where the microglia that are surrounding the tumor itself lose that motility. I'm wondering, this is somewhat of a piggyback, I guess, on Dr. Betterson's question, but early on, are there effects in the early tumor stages where microglial motility is impacted such that they aren't able to get to the sites of the early tumor cells to even have an effect? I mean, separating motility from their phagocytic ability. Is there any work that you all have seen with that?
So that's a very good question, Trevor. So it's interesting because these cells are phagocytic cells. It's a good point. And you will see some of them in the pseudopalisading necrotic areas, phagocytic cells. The thing about GBM is these cells are your own cells, live cells, and your macrophages won't phagocyte those live cells, right? So this is why they don't particularly lose their phagocytic ability. It's just there's no dead cell there to phagocyte those. But if you look at pseudopalisading necrosis and stuff, you can definitely see microglia phagocytosing the debris. It's just like it's good that they don't eat your live cells, because we would be in trouble if they start phagocyting your live cells, and your tumor cells are live cells. So, but if you give an injury or if you give a radiation high dose and you kill tumor cells, you can see microglia phagocytosing them. It's just in an opposite context. When tumor cell is alive, they don't know what to do with it, and they tumors hijack mechanisms to communicate with them, and they feed back to tumor cells. Like one of that is the IL-1, which usually is very well controlled in CNS because it's toxic for neurons.
Is it that early? Are there early signals from the tumor to prevent the microglial motility even, or is that what was leading into the microglial stasis that you were observing?
So we looked at the earliest stage. And earliest stage, we still see microglia in the peritumoral areas. And we see, even if we have a smallest tumor nodule, we see these cells coming from macrophages, monocytes coming from circulation. And they are mobile. They are mobile when they are in a progenitor state, because monocyte is not a macrophage. It's a progenitor cell. And this is why it's in circulation and it's very mobile there. But the moment they touch the tissue, they become like more stationary cells. This is why you have in tissues macrophages, not monocytes, and other way around in circulation. So I think it's the only way to target their movement is just to block their infiltration, because the moment they infiltrate, the differentiation process into macrophages and becoming a stationary cell is pretty fast and dynamic.
Thank you so much. That's great.
Thank you. Great question.
Hey, thank you so much again for the really interesting talk. I'm Alex, one of the third-year residents. Just had a question. So we know that IL-1 affects eventual tumor progression, but how is that related to the generalized immune response? Once, say, for example, if we downregulate IL-1, do we have a generalized downregulation in the immune response, or is there some type of compensatory immune reaction? For example, is NF-kappa B upregulated, or is there some compensatory change in the tumor microenvironment that we can potentially use as a target?
So that's a very good question. So yes, so we did, in IL-1 deficient mice, actually, IL-1 regulates NF-kappa B pathway activation. It's a known downstream target of IL-1. And then in turn, NF-kappa B drives IL-1, increases IL-1 expression transcriptionally. So it's a very nice circle there with NF-kappa B pathway. So when we, in IL-1 deficient mice, when we look at the immune system in general, not only in innate immune infiltrates, but so what we see there, we see increased infiltration of T cells, and we see less exhausted T cells in IL-1 deficient mice. So when we look at the PD-L1 expression levels, which is mainly in these tumors in myeloid cells. And when we look at the PD-L1, and we take the BMDM cultures and we stimulate them with IL-1, we upregulate PD-L1 expression level, which is not surprising that in knockout mice, we have less exhausted PD-L1 positive T cells there. I think we're finishing manuscript and actually writing that's probably there is a rationale to combine checkpoint inhibitors with IL-1 anti-IL-1 antibody, which actually very recently was shown to be very successful in the breast cancer models.
Great, thank you so much.
Dolores, I had a related question to that.
Yes.
To what you just said, which is that these tumor microenvironment and immune studies are interesting in GBM, and I wonder how generalizable they are to just other high-grade tumors. Does the brain's immune microenvironment react to all high-grade tumors the same way, or have we seen studies that show that different tumors are handled differently?
Yes.
More to the point of, could there be a more universal treatment for many different types of high-grade tumors?
So that's a great question, Peter. Let me just see if I got it right. So you mean high-grade tumors beyond glioblastoma in CNS, or just outside CNS?
Glioblastoma, ATRT, metastases, these are all, aggressive, poorly differentiated tumors.
Yeah, very good question. So even within the GBM, their activation is different. Their composition is different, whether we drive the tumor by PDGF overexpression in mice or we do by NF1 silencing, you almost feel you got two different tumor types. Their composition is different, their expression profile is different. The same they see when they look at the immune profile in human GBM, they say, well, macrophage signature genes are enriched in mesenchymal. But we did nail down in a more details on composition. And you see in a PDGF-driven tumors, your TAMs mainly constitute up from microglia 15%, 85% from monocytes, and you have only 2-3% neutrophil infiltration there. You go to mesenchymal GBM, which is NF1 driven, there you have much higher number of neutrophil infiltration compared to monocytes. You look at another high-grade tumors, if you look tumors in hemisphere in kids and DIPG, their immune profile is completely different. So I think there are differences. Within even CNS, there's a region-specific differences where the tumor form can affect. There's then oncogenic driver mutation effects there, that depending which driver is predominant there, is going to be the difference. So this is why it's been challenging to target these cells, not only in CNS tumors, but also there's no macrophage targeted therapy outside CNS tumors working either. So they've been targeting CCL2 and it hasn't been successful. Because you target CCL2, then there is CCL7, 8, and 12, and they compensate the function. So I think it should be, it's that not easy task to target them.
So a common target is going to be unlikely.
Mm-hmm.
Yeah. All right. Well, it's a little after 9 o'clock. Thank you so much for your talk today, and again, spurred interesting discussion.